Shalom לכולם, ברוכים הבאים. Welcome to our, so I will start in Hebrew and then remember I should be speaking in English. So welcome back everyone to our seminar series. And I'm very happy to see everyone here again. I will ask everyone to be kind and open their cameras so we can all see each other and get on equal ground. We have a special guest today, Professor Rony Paz from the Weizmann Institute, who is as good and friendly a neuroscientist as they come. Rony did his PhD in Jerusalem, at ICNC, and then did a postdoctoral work at, as a Fulbright scholar at Rutgers. And after that, he came back to the Weizmann Institute, first as an alone scholar, and then as an associate and full professor. And now he's the, are you still the head of neurobiology, Rony?
Sorry?
Are you still the head of the neurobiology department?
Unfortunately, yes.
So until he can get rid of that, he is still the head of the neurobiology department. And he does very, very exciting and high-profile work in primates and human primates. And I think his work is very, very relevant to the kind of stuff we're interested here at Gonda. So Rony, thank you so much for coming, and the stage is yours.
Thanks, Roy, for inviting. It's great to be here, and it's great to see all the good old friends as well as new faces. So I'll start right away. And I mean, the lab has been going in different directions in recent years. And I'll talk today, hopefully I'll get to talk about two different projects. I think the main one of them is going to be shared coding for value and social information in the primate amygdala, which was published 2 months ago or so. And if I had the time at the end, then I'll talk about what Roy really wants me to talk about, which is another paper published a few months ago with humans and fMRI, psychophysics and fMRI. And this one, shared coding, is going to be a work in electrophysiology in behaving monkeys. So I'll start from the second one, and we'll see how much time we have at the end. And if we have enough time, I'll try to summarize the first project as well. So, so the title is Shared Coding for Value and Social Information in the Primate Amygdala, and the person who did it all is, do you see my cursor? You do, right? Okay. Is Raviv Briluk right here. He's in charge of everything in this project. And so I'm not going to give a full introduction to the amygdala. I hope most of you are kind of familiar with it. But I'll just remind you the few things that are going to be highly relevant for our hypothesis here and then for the results. So amygdala responds, is known to respond for any kind of emotions and any kind of affective with an A stimuli, both for appetitive and for aversive reinforcers, punishment. It's a set of nuclei located in the medial temporal lobe, approximately right here. And of course, every person has his own, as you already know, every neuroscientist has his own favorite brain function. So that's mine. And there's a lot to do there in terms of understanding how we learn from emotions and from reward and punishment. It also, besides of responding to appetitive and aversive stimuli, which is across all species from humans in PET and fMRI studies and in animals with electrophysiology, imaging, any kind of method you choose, the amygdala also plays an important role in associating a conditional stimulus, Pavlovian kind of learning, with an unconditioned stimulus, so CS to US association. It's actually involved in more complex learning as well, but that's kind of the main work was done in this area. And it is well known to be involved in what we call emotional conditioning or affective conditioning, when the US is, again, appetitive or aversive and the CS is neutral. CS acquires the value of the US and the animal responds accordingly. I think the most famous paradigms are the one of classical fear conditioning in rodents. Most work was done in this region, identifying the specific circuit. I'm not going to go into it. This is obviously a cartoon, so the information about the CS, which can be a tone, can be auditory input, visual input, and the US, which is usually a food shock in the fear condition, but can be other kind of affective stimuli as well, are somehow connected, hypothesized by LTP, but as always with LTP, that's still to be completely proven. And at the end, the CS by itself can invoke via the amygdala and other regions. Obviously, the amygdala is not alone, can produce the conditioned response, etc. So the amygdala responds both to the US, to any kind of appetitive and aversive stimuli, but it also, after learning, and again, I'm emphasizing this because on one hand, we know that for many years, but it's going to be really relevant to the study we're going to do here and for the hypothesis. After learning, it also responds to encodes in single neurons for the expectation for valence. So the response to the CS changes in the amygdala after the CS was conditioned with a US of value. So remember these two aspects. One, amygdala responds to appetitive and aversive stimuli. Second, amygdala responds to learned CS after it was conditioned so it's kind of expectation for the valence. That's going to be the interpretation I'm going to use here. Yeah, so these are the two things to emphasize. On the other hand, so it's a completely different set of experiments, if you'd like. There's, I would say, a newer line of studies, but still already well established, that the amygdala is highly involved in anything that involves eye gaze. Obviously, this is done more in primates. There start to be some work in rodents as well, which is obviously harder and different. But in primates, it's well established. So these are one of the earlier studies showing responses in the amygdala for eye contact. Every time I say eye gaze, I mean when you look direct into the eye of someone, his amygdala is likely to respond more actively. That's shown in several paradigms. This is another famous study with, as there is HM for the hippocampus, there's an SM for the amygdala. It's this one patient who had her amygdala removed during epilepsy years ago. And Ralph Adolphs very nicely says in every lecture that thanks to her, he has a career, and that's approximately true. He published many studies with her and other patients, of course, but he showed nicely that the one thing that people without an amygdala or with lesion amygdala find it harder to do is to look in the eyes of someone in front of them. So they avoid eye gaze at all costs. And this is a study that we performed already 8 years ago. I think it was one of the few, if not the first one, of real-life interactions between two monkeys. There are many studies where you put picture, movie, whatever in front of a monkey. This is real-life interactions, and we just characterize with spike-based averaging the neurons responding to different eye movement in the other monkey face. So you can see high response to the eyes, both spike, and we show here both amygdala, and of course to the face. These two, the eyes and the face, are what constitute most of the facial expressions, also in humans, but in monkeys for sure. And therefore there's high activity representation for them in the amygdala. Another line of studies from Cadiz-Gotthard has shown directly that eye gaze invokes many more response in neurons. And there are other studies with lesions from NIMH groups, lesions in amygdala lesions in monkeys, again showing the importance to eye gaze and observation, understanding of facial expressions. So to summarize, amygdala highly responds to eye gaze of the other, of the person you're looking at or the peer you're looking at. And from these two, I want to develop the hypothesis that we started developing back then a few years ago. And so one first thing to notice is that eye gaze basically has valence, has value. And that is that direct gaze is considered a threat. I think if you think about almost all animals and species you can think of, direct gaze is a threat in most species, and especially in primates. Even in humans, I know some of you start to think about a mother and child, etc., etc. Just think about it for a second. If any time someone looks you directly in the eyes and is not someone you know very well for long, it's going to induce anxiety and you're going to feel highly uncomfortable. That's, I think, that's just intuitive. That's backed up by studies, of course, but I think it's also intuitive. So direct gaze is almost always considered a threat. In humans, again, it could be a more developed form of threat, but it's considered a threat in most species, and especially in primates. In contrast, averted gaze, again, almost always suggests submission. You can think about nature. You can also think about humans, linked to lack of confidence, shame, deceit, whatever. In animals, in nature, averted gaze usually always suggests some form of submission. But now, beside the fact that eye gaze has valence, it also predicts something about the outcome of the social encounter. This is some kind of hypothesis, but let me kind of justify it. It's kind of a conditioned response because it aids to expect and anticipate the next behavior. So direct eye gaze, again, in primates or in animals, because it's a threat, is a challenge and predicts danger for an attack or whatever. Averted gaze predicts a calm and permissive encounter and potentially rewarding, and ethology can give you hundreds of examples, all the way from mating, food sharing, offering. Most of them starts in monkeys and in many animals by averted gaze. And so I'm going to draw the parallel here to complete the hypothesis between the previous valence independent kind of coding and eye gaze, which is more of a social stimuli. So we have directed versus averted gaze. We have appetitive versus aversive, and you can see the analog I'm trying to draw here. And so aversive, I actually should have put it the other way around. Aversive should in some way parallel direct, and appetitive could parallel averted gaze. This is all hypothesis. I'm not saying this is justified for now. And another point is that there could be the two mechanisms that the amygdala is involved in: the response to the stimuli itself, the value, and the response to the predictor of the outcome after learning can also come about in eye gaze, because basically it has valence in itself and it can also be a predictor for the outcome. So this is just the kind of the showing you maybe how we thought during the first few months when Raviv started his PhD. How do we started this hypothesis and came to the idea of the work. So just to emphasize again, averted versus direct gaze might parallel appetitive to aversive stimuli, and it could be both for the US, response to the US, and response to a CS conditioned stimuli. So yeah, and another line of evidence, which I'm going to come to at the end, but that's kind of always interesting. I'm not sure it's convincing by itself, but it's interesting, is that many conditions that involve malfunction and perceiving and construing eye gaze, obviously autism and other conditions, anxiety disorders definitely, but autism as well, frequently include the anxiety and value-based aspects. So again, there's some, this is, I said, this is just another nice to have, but there's some analogy even here between the classical roles of the amygdala and the social roles, and the fact that they're comorbid in many such psychopathologies. Okay, so Raviv therefore formed the following hypotheses or questions. Is it the same circuitry that is used for direct versus averted gaze and for repetitive versus aversive stimuli? We know the reason this is an important question, if you like, is that there's what's called the social brain hypothesis, which posits and have been claimed by many that dedicated circuits have evolved to interpret social cues in advanced evolved primates. So that's why it's going to make sense to ask this: are these the same circuits or different circuits? And the similar question from what I showed before is that, is it same circuitry for direct averted gaze, and not only the stimuli itself, but also the CS, which I remind you is neutral at the beginning of learning, but acquires positive response. It can be in the same circuit, it can also be in a different circuit. So that's a question by itself. And again, is it the same circuitry and similar mechanism for the gaze in US? I mean, assuming the upper two are correct, is it similar for both the gaze US and gaze CS? I hope that's clear. If not, I think it'll be as I go along. So these are the hypotheses we started with. By the way, I don't know what you prefer, but you can stop me for questions if you like. I think we can manage it. So we wanted to test this in monkeys, which to me makes sense because they are evolved primates with direct and eye contact, and they have a highly evolved amygdala. And the question was how. So we adopted a paradigm which is kind of well established, called the human intruder test. It was invented by Ned Kaelin, who's a psychiatrist, and he used this paradigm actually in a similar way to how we're going to use it here, which is just there's a monkey and a human walks in and either looks at the monkey, looks away, and it's called the human intruder test. And he shows, he has hundreds of monkeys in a facility, and he does a lot of tests. He's mostly interested in pediatric development of anxiety. And so he shows that the HIT response, the response to a human intruder in infant babies, is an early phenotypic risk to develop anxiety psychopathology in monkeys. I won't go into all the details. It's linked to metabolic activity, functional connectivity, and even heritability, genetic heritability in expressed genes in the amygdala. And it's a line of work. We just thought it's well-established paradigm, which makes it very nice. And we add to it what he did in 1 or 2 papers, but not controlled enough, so we do it in a more controlled way, is that the intruder now has 2 controlled conditions. One is where he looks direct eye gaze, and the other is when there's no eye contact, when he looks away with a stable gaze towards something else. This is a shutter that opens and closes with the 1 millisecond response time. So it signals the beginning of a trial. Sorry. And the animal does not know whether when it's going to open, it's going to be an eye contact or no eye contact, and they are randomly interleaved and repeated, et cetera, et cetera. So we had the eye contact versus no eye contact. We're going to call this social trials, and there are 2 conditions in the social trials: eye contact versus no eye contact. And there's another kind of block that is interleaved. So you see in the bottom here just an example of a session. The human intruder test or session is interleaved with aversive blocks and reward blocks. So reward blocks are very simple. Every time the shutter opens, the monkey after a few seconds gets a reward, as you're all familiar with. And aversive is similar, but the monkey gets an air puff, an unpleasant air puff. It's not painful, but it's highly they don't like it. And those are different blocks, but they're interleaved across the session. So basically the idea here is very simple, is that, I'm not sure that this explains the idea, but we can examine within the same neurons, because these are all done within one session in a day. We can examine in the same neurons the response to eye contact. No, first the response to social versus reward air puff blocks. Second, the response to eye contact versus no eye contact. We can compare, which I'm basically summarizing the paper, almost the end result for you. We can compare eye contact and no eye contact to reward and air puff, as I said before. And we can also compare eye contact and no eye contact, the neural response, to the response to the CS, in this case, the shutter opens, which predicts the reward. In this case, the shutter opens, which predicts the air puff. So we can compare it both to the US and to the CS. So we kind of have all comparisons on the same neurons, which usually comes up to a big mess in the number of trials and analysis, but actually it came out okay here. So the nice thing is that they do hundreds of trials per day in this, so we have enough power for this. Okay, so just a few basic things about the behavior. There's going to be nothing too surprising here, I think. So just for the sake of definitions, the humans are all sit in front of the monkey. We know exactly where they are. They are filmed every day. It's the same region. It's all fixated, and we know exactly what is the face region. We're going to call it the face ROI, and we know exactly what are the eyes ROI of the person. And we also record and track eye movements of the monkey, so we can see exactly where the monkey is looking.
Rony, can I ask something?
Sure. Sure.
Is it the same person that they see every session, or is it a different person?
So that's an excellent question. And so in order to increase the interest and decrease even more the statistical power, again, turned out completely okay. But we had every day, we had 2-3 sessions of a familiar individual, which is the caregiver. We had 2-3 sessions of the same unfamiliar individual, which was basically one of the students in the lab that the monkey doesn't know at all, because this person doesn't work with this monkey or with monkeys at all. And we even had at the end, we even had monkeys that are as the intruder. I'm not going to tell you about it today. And your question is if I can guess where you're going, it's definitely something that we hypothesize on. Is there going to be a difference between a familiar and a non-familiar intruder? And this is still under analysis. It's much more delicate differences than what I'm going to show you today. Invite me again in a few months and I'll tell you if there is. So this is just what you'd expect. So all the human intruders, I say, are going to go to social, and all the reward or air puff is going to be called non-social. And this is just the time in a cumulative distribution function, just the time for the first time the monkey gaze into the eyes ROI. So the first time the monkey looks somewhere into this rectangle, that's the cumulative time plotted here. And obviously he looks much more to this. This could be, actually it's interesting that it's non-random, maybe because he's confused about the task, but there's definitely much more looking to the eyes, which is expected. This is just an example of one of one trial, the scheme, and these are the single trials. This is a single trial. This just jumps in, I don't remember how many milliseconds time, but you see the eye jump before the shutter opens, then the shutter opens, and he looks, and the monkey looks directly to the eye, and then he decides, usually he looks away, or actually that also makes sense. For social reasons, he looks after the eyes, he looks at the mouth, but I'm not going to talk about that. This is the overall density, overall trials, nothing, I mean, it's just showing that the paradigm kind of worked in that sense. By the way, it also shows that the monkey doesn't look before the shutter, which is going to be delicately important for controls later on, but yeah. So if we now take only the social trials and look at the eye contact versus no eye contact, I remind you these are randomly interleaved, so the monkey doesn't really know, and so when the shutter opens, he looks with the similar, exactly similar kind of time frame. He looks to the eyes region, and then he either looks away in no eye contact or stays there. This, I mean, this is actually a huge difference because it's average over all trials, etc. So it basically means that the monkey looks much, much more, stays with the eyes and for a longer time on the eyes of the intruder in eye contact versus no eye contact. And this just shows that for eyes ROI versus face ROI, that's the same kind of result. Another thing we did, which is to look at the actual expression on the monkey. I'm not going to talk a lot about it today because there's another paper which we hope to submit soon. We actually characterize exactly facial expressions. So there are facial, there's neutral expressions. This was the typical response to the eye contact and the response to the no eye contact. And you can see that there's, as I hypothesized from ethology in the beginning, it's kind of a more aggressive threat-related response to eye contact, much less to no eye contact. And this shows the difference when averaged over, I think it's the upper face or the lower face. In both cases, eye contact evokes more facial responses. We now do it with some more nicer automatic facial expression detector and lock neurons to it. I'm not going to talk about it today. And the other thing, again, just validating the behavior, is that the monkey indeed learned the reward versus error path, and reward, actually, there are no licks here, there should be licks here, but shown are the eyelid responses. And you can see that in reward, the eye stays open, and in the air path, obviously the eyes closes, and this is happens not only after the air path, but also as a preparatory signal. So it happens approximately 400 milliseconds before the air path comes, which means that the monkey actually knows that shutter open is going to predict an air path in this case. So we have basically everything we wanted from the behavior. We have responses both to the eye contact versus no eye contact, and to reward versus error path, both for the US and for the CS, for the expectation. We performed also a lot of autonomic measures, and not going to go too much into them, but there's a heart rate and the heart rate variability, which are both, heart rate variability is actually the more used measure. But what was nice here is that if you look at the response to the eye contact versus the no eye contact, it's kind of similar to the response to the air path versus the reward, again, suggesting, at this point, it's just suggesting that they have kind of similar direction or or valence. Okay, so we recorded every day in the amygdala and also in the anterior cingulate cortex. There's a reason for the cingulate cortex. I'm going to use it here mainly as a control, but cingulate cortex is also highly involved in primates in looking in affective stimuli and also in social responses. So it was even, I would say, an interesting control and not just some random brain region. And this is just showing that we know how to record in the amygdala. Most of our recordings are in the basolateral complex, the BLA complex of the amygdala, which is the basolateral and the lateral BL and LA nuclei of the amygdala. We almost do not record in the central amygdala, which could be interesting, but this, for our sake here, it's less important. And as you can expect, there are dozens of neurons that respond in both regions to I mean, these are just some examples. There can be tons of them. There's a whole zoo of cells that responds to the US itself, to the expectation, to both of them. Whatever you choose, I can find you a cell that responds to both the CS, the US of the reward, and so one color here is the ERPAF, the other is the reward. Trials within the same session. And so both the amygdala and the cingulate respond and discriminate valence in both CS-related and US-related responses. This is validation of many studies. So this is how many neurons respond to the CS ERPAF, to the CS reward in the ACC and the amygdala. And this is how many discriminate between ERPAF and reward. And this is the same for the US. It's actually kind of surprising that US here is a little bit low, but there's no big difference, nothing too interesting in this result. Both regions know how to discriminate valence, and both regions respond to both CS and US. And if we now look at eye contact versus no eye contact, again, I remind you, these are different conditions within the same block, interleaved and randomized. Then again, we can find all kinds of cells, but the important thing here is that if we try to see how many cells discriminate between the human intruder test and then social and non-social blocks, then the amygdala is much higher than the cingulate cortex. And what's even more interesting, at least for us, is that the amygdala discriminates. I mean, let me phrase it differently. There's a significant number of neurons in the amygdala that can discriminate between eye contact and no eye contact. Whereas in the ACC, there are few, but it's at the chance level, if at all. So the amygdala can discriminate both social versus no social, not surprising. The ACC can as well, also not surprising. This is a bit surprising that the ACC does not discriminate gaze, and the fact that the amygdala does will be important in a second. Okay, let's skip this. One thing to remember, which I'm going to come back at the end, is that if you look at the overlap between gaze and valence, which is an analysis I don't like too much, and I'll say at the end why exactly I don't like it too much, is that if you look at the overlap, then the overlap is highly minimal. It seems that there are different circuits that if you like, per our hypothesis, there are different circuits in the amygdala that code for gaze and for CS or US. So the proportion of shared neurons is really minimal. So that actually seems like kind of a bummer for our hypothesis. But if you then take the decoding approach, which is on one hand all too popular these days, on the other, it tells you something about what's going on in the network, and that's why it's so nicely used in many studies. So this is done using population response vectors, training linear decoders. Won't go into the techniques. It's standard decoding approaches, and it really doesn't matter what kind of approach you would choose for this. So if you try to decode CS valence by building population response vectors, then you get a nice accuracy for both from amygdala neurons and from ACC neurons. If you try to decode US valence, appetitive or aversive, again, you get nice decoding accuracy. And as shown by the limited number of neurons responding in the ACC to gaze, to eye contact versus no eye contact, then you cannot decode it from the population as well. But in the amygdala, again, I'm just going back to this. This is basically reflecting this. And then from the amygdala, you can definitely decode whether it was an eye contact or a no eye contact trial. So the question is now, is there shared coding for gaze and valence? And on one hand, I kind of gave an initial answer, which is likely not, because the overlap is minimal. But there's a more general way to test this. And this is to take the decoder, train it on one condition and test on the other. Again, this is, I would say, maybe too popular these days, but it gives a very good approximation of the simple question, is there information in the population about the other condition? So basically that is what we do. We train the decoder to distinguish between air puff and reward, and we test it on eye contact and no eye contact, or vice versa. So when we do this, then you get potentially the result in our hypothesis, which is there is kind of a shared circuitry or shared coding, some kind of shared mechanism that if you train on the, now I can't remember if it's trained on conditioning and tested on human or vice versa, but it doesn't matter so much. It's almost symmetric. So you train on one and test on the other, and in the amygdala time from the, it's trained on human intruder because you can see that it's tested on the outcome of the US. So it's trained on the human intruder, tested on the classical conditioning, and you see that after some time you get a nice above chance accuracy, although it was trained completely only on human intruder trials in discriminating eye contact and no eye contact. Independently on the anterior cingulate cortex, there seems to be no signal at all. This is a number of neurons. It's actually, I'm not going to talk too much about it, but it actually seems to be linear, which has a lot of implication we can talk about. But since we did not record many neurons here simultaneously, it's only 4 to 5, we're limited in seeing whether this is really a linear because of independency between the neurons or something deeper than this. Okay, so that's training on human intruder, testing on the US, appetitive versus aversive. What happens if you look at the CS related responses? Well, you get the same, maybe even a nicer kind of accuracy. So you train on the human intruder, discriminating eye contact to no eye contact, and you get a much above chance level accuracy in discriminating CS that was conditioned. So here it's just shutter open that was conditioned with air path, and shutter open that was conditioned with reward. And again, nothing, almost nothing for the ACC, and it increases linearly with the number of neurons. So the first approximation answer is that yes, there's shared coding for eye gaze and US valence, and moreover, there's a shared coding for eye gaze and CS. And even more, eye contact to no eye contact is somewhat similar to aversive to appetitive valence. And that's simply because of the direction of the decoding. So here you get an improved accuracy And this is just trivial, but if you train it on, take the NEC to EC and try to do aversive to appetitive, again, it's completely trivial. It's symmetric to the previous one. This should be enough for you to say that the direction is the same, but it means that in some way eye contact shares some coding similarity to aversive, and no eye contact shares some coding similarities to appetitive. And so this was kind of our hypothesis, and that was nice to find. The question that remains is it the same coding mechanism? Is it that eye contact to no eye contact is like aversive to appetitive in the same way for CS as it is for US, which does not have to be the case at all. The fact that the CS is a completely different thing. It's expectation to the US, it's acquired value. US is, if you like, innate value. Okay, so there could be, in principle, two groups of mechanism of shared coding. This is very rough. I mean, there could be more delicate subgroups of these coding for sure. But if we divide them into two large groups, then one of them would be the overall activity, which just means that neurons either, some group of neurons respond more vigorously to EC than to NEC, and another groups of neurons respond more vigorously to aversive versus appetitive, but they're not the same neurons and they're not correlated across the response. And so the population as a whole responds similarly, and we can decode, but on a neuron level, on a single neuron level, they're not correlated. The other is what's called correlated selectivity. And again, it's a very simple notion, been with us for, I don't know what, 30, 40, Moshe can testify, maybe even 50 or 60 years. But correlated selectivity just means that the same neuron responds similarly in the same direction to EC, NEC, and reward AirPuff. So if you plot this on a 3-neuron paradigm, just as an example, this would be different neurons raising their response, but still the same separatory surface. It wouldn't be the same as if trained over the other paradigm, but by and large, it would do a good job if you use the one here, just because, again, by and large, there's a whole similar population activity. Whereas here, you'll still get shared coding and you can decode, but it's based on a deeper kind of fact, and that is that the same separatory surface would be almost similar, almost parallel in the two different conditions, in the human intruder and in the classical conditioning. Okay, so we want to test this, and obviously want to test if one of them plays for the CS, the other for the US, etc. And there are several ways to test this. I'm going to show all show the same thing, but I'm going to show several approaches. So one thing is to look in single cells, and this is just, so you look at all the trials of the no eye contact, all trials of the eye contact, you fit some kind of a linear fit. You do the same for AirPuff and reward. And now you're going to look at the coefficient for both of them and simply see they are correlated between the conditions. And the answer is they are highly correlated for the amygdala in expectation. So it basically means that the situation looks like what I'm showing you here. It's the same kind of linear fit with the same slope. But that's not the case if you look for the US, the outcome. So this is CS expectation, and this is outcome US. So it's not the case for the US, and it's also definitely not the case for ACC, which shouldn't be surprising at this point. What's more important now is the difference between the CS and the US, and the fact that these are correlated. The slope of single cells is correlated across across the two different conditions. So that's the first evidence that they have different mechanisms. Another way is to look at that in all cells together. Just do multiply the vectors, and then shuffle and see how it looks in real neurons versus in shuffle neurons. So if you look at the outcome, then which is also really yeah. Then for the outcome, then the mean of the actual neurons is not different than the shuffled ones. Actually, the standard deviation is much, much bigger, and that's because that makes sense. It doesn't matter what kind of shuffle you'll do, because it relies on the whole population approach, on the whole population activity, you'll get much more standard deviation. I hope that's clear. On the other hand, for the expectation, just like I showed you here that single neurons are correlated, so if you look at you get a nice decoding from real neurons, and if you shuffle the neurons, that is, you take the activity of one neuron in eye contact versus no eye contact and match it with the activity of another neuron from aversive to appetitive, you ruin completely your ability to decode. So that means that the identity of the neurons is meaningful and basically reflects what you see here. They're not independent results. One of them is tightly related to the other. Another way to show this is done on a single level, a single cell level, and this is taking them as a vector, but you can also do population decoding. So you collect from all the days, sum them up, and try to fit a hyperplane, as I showed, as shown here on free neurons. That's done on the almost I don't remember the numbers now, but approximately 200 neurons in each region from 2 monkeys. And so if you do that, then the cosine between the angles of the two separators are either close to 0, which says they are perpendicular, or they are farther from 0, which says they are not perpendicular. I'm saying here parallel, it obviously doesn't mean they're parallel, it just means that they're not perpendicular and more towards one another. So in the ACC, there's no relationship, but that shouldn't be surprising because there's no real coding. If you look at the US outcome, then they are almost perpendicular. Again, showing there's no relationship between the two, kind of like, I'll get to it later. And in the amygdala, in the amygdala and the expectation, they are significantly different from perpendicular, which means they are somewhat more similar. The two planes are somewhat more, two separator planes are somewhat more similar. Another final way to do it is to again do the decoding and shuffle the neurons. And so for the outcome, you have a decoding which stays the same even if you shuffle the neurons, as I said earlier, because the order doesn't matter, it matters how much the activity. But if you shuffle the order of the neurons for the expectation, for the CS, you completely kill your ability to decode properly the other condition, because again, the neurons are no longer correlated because you shuffle their identity. So yeah, we can skip this. So basically, there are good evidence, in my view at least, to having two different mechanisms. Again, there's nothing new here about the word mechanisms. It's not a new mechanism of coding. These are classical, well-established, I mean, high groups of coding possibilities. But the important thing here is that they are very much different between eye contact to no eye contact and appetitive to aversive. If you look at the eye contact to no eye contact and you compare it to the US, to the outcome, AirPA for reward, then it's an overall activity kind of a mechanism, but random identity of the neurons. If you look at eye contact, no eye contact, and compare it to ERPAF reward, but to the CS, it's CS, ERPAF, CS reward, which is a neutral stimuli that just predicts them, then you get correlated selectivity and the same neurons responds at least in the same direction to both in both these trials, which, I mean, okay, let's get to the interpretations. One can claim whether, one can ask whether this is a more advanced evolutionary mechanism. Does it require something more from the network, some more some different wiring, and actually can go either way. We can discuss it at the end if you want. One thing which I mentioned earlier, and I'm just going to conclude this project with this, is that this is kind of a small point, is that I kind of showed you somewhere in the middle that there's very little overlap between the neurons that code for gaze and for CS or US. And I even said at this point we were kind of bummed because our hypotheses didn't look too good. And I think there's nothing really deep about that. It just means that we're thresholding our populations too much and using significance as kind of a sacred number, which is, and that's, I guess I'm preaching to the choir here, but you still see tons of these studies, and the question whether we should use significance or more gradual analog-like versions, which decoding, for example, is a good example to circumvent the problem of significance. And so the question is, why do we get so nice decoding even though there's little overlap? And the point is simple. You're always limited by the power of your the number of trials you made. And if you look at significance of any kind of formal test, you lose all the neurons that are basically can code in the same direction. This can be either because just because you didn't have enough power, or just because the neurons don't respond vigorously enough, and the actual brain relies on more sophisticated readouts, I guess. So just to make sure this is the case, if you don't care about significance and you just take any kind of an index, which is the response to the response to CS minus appetitive minus aversive divided by appetitive plus aversive, and you do that once for the, who came up with the name SIV and SIG? I now can't remember which is which, but that doesn't matter. And if you do that, you devise an index of directionality for the aversive appetitive and for eye contact, no eye contact. And you can see that even though many of these neurons do not have a significant response, the significant would be approximately here and here and here and here, but you still get a nice correlation between them over the whole population. So you can definitely get this decoding ability even when there's minimal overlap with just formal significance. Again, it can be either because the actual activity of the neurons or because you're just underpowered in statistics. Okay, so I'm gonna summarize this part quickly. So we think that direct and averted eye gaze share the similar circuitry or similar coding scheme with valence, simply because we can decode one from the other and vice versa. And there's good evidence that it goes in the same direction. So direct to averted gaze corresponds to aversive to reward. And it's harder to say exactly what are the similarities in the coding, but at least in terms of decoding and looking at across the population, it looks like they are in similar direction. And this goes nicely with our initial hypothesis that A direct gaze is more related to threat, etc. And averted gaze kind of more related to submission, which means reward in the animal world in general. And maybe a more important thing and more interesting for us was that eye gaze does not only share coding with valence, it also shares coding with expectancy for outcome. And this is not trivial because I won't go into describing all the amygdala literature, but it doesn't seem to be the same neurons that code for CS and US. This is still a big open debate in amygdala literature. I can't think of there are several Nature papers that claim this way or the other. None of them is convincing, but at least here it seems that the eye gaze shares coding scheme with expectancy for outcome. And there are two different mechanisms. Actually, the names are different. It's either overall activity for gaze in US and what we call correlated sensitivity for gaze and CS. Okay, so these are the findings basically. And now we can say at least my interpretation from it. So one is that processing and interpreting of eye gaze likely evolved from circuits that are shared with non-social elements. That is a well-known phenomenon in evolution of looking at repurposing different circuits, not just in the brain, obviously in all organs, but here it also makes sense. Again, this is kind of different from a common social brain hypothesis that posits that social circuits that look at eye gaze, especially in primates, has evolved in completely dedicated circuits. And obviously, I'm not trying to say that there aren't also additional circuits that look at more complex social interactions, but still that the amygdala circuit at least can code for both of them at the same time. Yeah, so we know today that the amygdala evolved with social interactions, and such responses might enable complex social processes, learning, social-based decision-making. These are all related in my view, because if eye gaze is related to valence, then I think it's kind of self-explanatory how it underlies learning. And then finally, the complexity of neurodevelopmental disorder, this is obviously an interpretation at this point, and especially at autism. They all show that simple aspects of gaze processing are disrupted, and understanding of the other intentions, emotions, and actions are compromised, all accompanied with abnormal amygdala function, and all has huge anxiety, anxiety with comorbidity between them, which in my view, again, very straightforward. If you have a malfunction in some circuit and this circuit codes for two different things, then you'd likely have abnormalities in the two functions. So autism and anxiety, and social anxiety is almost kind of the definition of what we do in the, I mean, of what comes out of the study, at least in my view. I think that's it. Maybe, yeah, that's it for the first part. Wow, took me a full hour. So I don't know, your call. Do you want to ask questions, or do you want me to just show the final slide and ask questions?
Sure.
Okay. So sorry, Roy, I knew it always takes more time than I thought, but I urge you to read this paper, which is short and cute, although it's not easy to understand. Even for me, it takes time now to read it, but we basically found, and I'll say one word about it because it's kind of the same line of thinking, I would say, but in a different system altogether, is prediction errors, which I assume most of you know what prediction errors are are coded by dopaminergic neurons and shown a lot in striatum responses. And on the other hand, time estimation is also highly attributed to striatal activity. And so we simply ask, because the two functions, and it's a completely two different lines of studies. One's about time estimation, sub-second time estimation, the other about prediction errors. And we simply ask if they are both represented in the striatum, then they might interact functionally. And we did some study that looked at that and basically found that you have a complex interaction between prediction error and how it biases time perception. So every time you're expected for good or for worse, it actually means that you likely estimated differently the time before it. I think it has a lot of interesting implications for learning, etc. But yeah, and we showed this interaction actually by modeling an fMRI. We showed that it actually tightly related to the striatum, so kind of validating our hypothesis. Okay, so Raviv did almost single-handedly the work of the first paper. Ido did almost single-handedly the work of the second paper, which I didn't get to talk too much. And these are all the people that helped a lot during Daphna and Anna and Yossi. And Eilat helped Raviv, and Chris worked with Ido, and many others. That's it. Thanks.
Thank you. Okay, so thanks a lot, Rony, and we have time for some questions. Okay, so first of all, I'll take my prerogative and start with the first question. So in a way, and this is something maybe you can answer from your own data, Rony, you can think of the gaze aversion as also as a kind of conditioned stimulus. So if you look me in the eyes and I slap you in the face a couple of times, then this is something like the animals do sometimes that you will learn. And then it's not so surprising that in amygdala it's encoded in a similar way. But then this you can maybe, if you recorded during the learning phase or considering how your monkeys were raised, maybe you can actually answer this from your own data.
Yeah, so first I agree, and that was our hypothesis. I mean, you're asking correctly. Is our hypothesis that eye gaze is expectation in the same way that a CS is being learned, or is it expectation from evolution, some hardwire, I don't know what? Obviously, we cannot answer this here completely. I think both of them are as interesting for me. The only way I can think of to do it, and I'll tell you in a second what we're doing now, is we try to do it is by kind of training. I mean, so there are two ways. One is what I hinted to. First, okay, does it make sense? I mean, yeah, I agree that eye contact is a CS. That was our hypothesis. And it can be a CS due to two mechanisms, either learning or just because it was wired over evolution to be so. Both will be interesting. How to distinguish between them is definitely not easy. We could try and condition an eye contact with an air puff or a reward. It's, if you think about it for some time, it's gonna really make interpretation complex, but it can be, but we actually do it now in humans with neurosurgeons and trying it just easier in humans than in monkeys. So this is the work of Arik down here, just starting to see if we can actually change the value of eye gaze, either because it's being conditioned or because, and that also relates a little bit to what Dana asked me early on, and I still don't have a good answer. Is it, I mean, would different people with different value have, but that doesn't answer the CSUS dichotomy, it answers just a valence dichotomy. So we need to change it and see if it can be changed, conditioned. That's a good point.
But actually, I think maybe if your monkeys were reared without any social interaction, they would never be slapped by, they would never be rewarded or trained to respond in a different way to gaze. So in that sense, if they still have these responses, this would be an innate response, right?
So, I mean, again, you're asking me to hypothesize because I can either claim my view whether the responses are innate or whether such social responses are innate or being learned during development over learning. I'll say I'm not that of an expert. I think there are bigger experts than me there in development, but I think this is not even clear. It's almost like the Chomsky problem. Nobody can demonstrate whether it's this way or the other. So I don't know. Rearing monkeys without social interaction is not something that we can do, not here and probably not anywhere these days. Maybe maybe if, you know, can be tested in rodent. I don't know, but it's a hard question to test.
Okay, thanks.
Questions? Yes, Rony?
Yeah.
Okay. So I'm wondering, do you think that the amygdala actually drives the gaze, or is it a different area in the brain that is responsible for the gaze? And
yeah, it's a good point. So first, let me go back to actually our studies from 2012 and where is it? Oh, it's here. No. So we tried to address it a bit. Come on. Okay. Yeah. We tried to address it a bit here in a more careful analysis. It was a different kind of, actually no part. It was free viewing. And, you know, these things are never, you know, as well as I do, they're looking at whether the change is pre-spike or post-spike is not a causal demonstration of either way. But you can see there's some, you can see that the distribution here is much higher for post-spike. So that means that it seems like the spikes are actually predicting the, well, either predicting or driving. I just don't know at this point. The actual change in the eye region. There is no evidence for stimulating the amygdala. It has been tried, but there's absolutely no evidence of stimulating the amygdala and causing movement of, it's not like frontal eye fields or things like that. It can bias eye movements a bit, but even the bias is not that strong. So I would say it doesn't drive, definitely doesn't drive directly in the classical way motor people or whatever I used to think about it. And it definitely reflects, in my view, and this was the point of this paper, it likely receives afferents from eye commands. That was actually the point in this paper. But just to be clear, here in our, what I'm looking at is responses that are to the eye gaze of the other. So it doesn't relate to my own, and the responses do not reflect. There are several controls there that I did not mention, do not reflect at all the movement of your own eyes in this case. It just, the responses that I'm showing here reflect only whether the other is looking directly at you or looking away from you.
So it's basically could be the emotional response only.
That's a good point, and the answer is no. It's not the emotional response per se. I mean, it is the emotional response if it's similar. Yeah, if you have a similar emotional response to direct eye gaze and aversive air path, then definitely yes, you cannot rule this out. But sure. But the response is during the time. It's not a developed response afterwards. Okay, so it's not the response to all the facial expressions that develop later. It's a response to the first observation of the averted versus directed eye gaze. So it's not a reflection. Let me put it this way. It can be a reflection of the emotional response. It's not a reflection. It's not a correlate of the actual emotional reactions. Okay.
Yeah, I think we'll talk about it separately, I think.
Okay, but look at the paper. I mean, it's obviously was a big deal, and we did several controls there to show that it's not. Let me jump. I think I have some slides at the end. Yeah.
Yeah, because we respond to many things without really identifying them, right? It's just that the initial impression that we receive sometimes is sufficient to get an emotional response that of, like, you really need the fight or flight thing to be quick, right?
Sure. And that's exactly my interpretation. The only point is that it's not, that I want to make clear, is that it's not of the fight or of the flight. It's of the response that leads to that. And that's fine with me. A direct eye gaze is like an aversive stimuli in amygdala circuits. It's not the fact that I later run away from both. Okay?
Yeah, great, thanks.
Good. Any final question? Because we're running a bit late.
I have a quick question in regards to the gazing. I'm sorry, I don't have video right now, but when you were saying about the gaze, did anybody, or I'm curious to see if you guys did any differences between gazes, because obviously depending on the emotion that precedes the gaze, I don't know if anybody has kids, but you could stare at one and say nothing and look straight and they could know exactly what you're saying, if it's good, bad. And I'm wondering if that had any factors of, you know, the type of expressions with the amount of muscles we have in our eyes, and also why, if it's not dangerous, if was this ever tested sort of on humans, to going through trauma and different elements that could lead to different eye gazes, and obviously maybe coming from different sources.
Yeah, so I assume you know more than me about these things. I can just hypothesize. I don't know a lot about kids and different, how miniature scale can we really detect different eye gaze. I'll be bold without knowing enough to say that we probably overestimate our ability to differentiate between different eye gazes. But again, I'm sure you know more than me, and there probably might be studies that actually show that we can. It's definitely not at the level of this study. I mean, you know, first, I mean, I'll be happy to think of ideas, but I wouldn't know how to instruct a person to do different eye gaze so that the monkey would use the simplest thing of direct gaze or averted gaze. And that was a signal we could clearly identify. If you have any ideas how to do more miniature scale types of gaze, I'll be happy to hear about it.
I'll contact you.
What? Sorry?
I said I'll contact you. I have lots of ideas.
I mean, the reason, and what I mean, if you'd ask me to do monkeys, I'll say I'm not sure it's going to work. But we're now actually doing it, as I said, almost same paradigm. But we're trying to condition eye gaze with aversive, repetitive, and also play with familiar, unfamiliar, because we know more in humans what it means than in monkeys. So we do it now while recording in the amygdala of epileptic patients, and you know, it's a long process. But if you have any idea for manipulations in humans, it might work better.
I will definitely reach out. And then if you have a quick sec, speaking of the same type of differences, the gazes, when there's no gaze, I'm wondering if they're gazing at something else versus being blind per se, which would, you know, maybe trigger other things. If different, what are they looking at? And does that sort of instill something else that can cause reaction? Obviously, animals oftentimes have senses outside of just visual.
all other conditions were kind of identical here. So, I mean, there are obviously a lot of function cues, etc. But that's why the eye contact, no eye contact is completely balanced, and there's no difference in all other. So everything is completely identical, beside the fact, I mean, everything is completely identical, beside the fact that you either look directly or look away. So we made sure of that. And the monkey himself looked different. I mean, that would require a sub-analysis, whether he then looks like like you saw in the example, look at the face or looks other. That could be interesting, but we didn't look at that yet.
Okay, just curious. Thank you so much. It was a great lecture. Thank you.
Okay. So let's all thank Rony again for a fantastic talk.
Thank you for inviting.
And I'll have to invite you again. You still owe me the paper I wanted to hear.
Okay, sure.
But then by then you'll have another 2 Nature Neurosciences. We'll probably talk about them.
With pleasure. I mean, going to Bar-Ilan seemed, I mean, you know, we go easier to New York than to Bar-Ilan or to Weizmann in the pre-corona days. So now we can do it by Zoom, which is great.
Fantastic. So thanks, Rony, and thanks, everyone. Have a good week and keep safe.
Thanks. Bye-bye.